For decades, scientists who are, trying to study Alzheimer's disease, they've been forced into this really, really frustrating compromise.
Yeah, a massive compromise, honestly.
Right. It's like you can either map the whole brain and see almost no detail, or you can take a piece of that brain and basically blend it into a biological smoothie to study the individual cells.
Exactly. But when you do that, you lose any idea of where those cells actually came from.
Yeah, you destroy the map to understand the territory. It's wild. But today, we're looking at a brand new technology that finally lets us do both.
And what it's revealing is it's nothing short of incredible, really.
Oh, absolutely. We're talking about microscopic hazmat teams swarming toxic spills right inside the human mind.
It completely changes our understanding of the disease. I mean, we are moving from looking at Alzheimer's as this generalized brain fog to seeing it as highly localized guerrilla warfare at the cellular level.
Welcome to another deep dive. Today, we are immersing ourselves in a really groundbreaking paper that was recently published in Nature Neuroscience. And, for you listening, our mission today is to examine how these researchers used a complex new imaging technology, it's called CODEX-CNS, to look at the frontal cortex of both Alzheimer's patients and healthy controls.
Yeah, we're going to look at exactly how this new camera works.
Right, and the staggering cell shapes it revealed and how it caught a highly specific immune cell red-handed at the scene of the crime. Okay, let's unpack this. Where do we even start with a leap forward like this?
Well, you really have to start with the technology itself, because without it, we're still just staring at the smoothie.
Right.
To really grasp the magnitude of CODEX-CNS, we need to appreciate the wall that neuroscientists have been banging their heads against for years.
Right, single-cell RNA sequencing, which it's been the gold standard for a while now.
Oh, absolutely. And it is a brilliant tool for understanding the genetic recipe of tissue.
Yeah.
But the limitation is massive. As you mentioned, it's like putting a chunk of the brain into a blender.
Yeah, you run the analysis on that blended liquid,
Yeah.
and you know exactly what ingredients were in the cup. Like you had an abundance of a specific mutated gene or a certain immune cell.
But you have completely destroyed the neighborhood.
Exactly. You have no idea if cell A was sitting right next to cell B, or if they were on, completely opposite sides of the tissue.
Exactly. You strip away the spatial context, which, in an organ as intricately wired as the human brain, is basically throwing away half the information.
Yeah.
On the other side of the spectrum, we've had traditional immunofluorescence. This is where you keep the tissue perfectly intact, and you use glowing antibodies to tag specific proteins.
So you don't blend anything.
Right, no blending. But the problem there is bandwidth. You can only look at 2, maybe 3 proteins at the exact same time before the glowing colors they bleed into each other and become this unreadable blur.
So you either get deep detail with no map or a map with almost no detail, which brings us to the real breakthrough in this paper, which is CODEX-CNS. So CODEX stands for co-detection by indexing, and the CNS is for the central nervous system. How does this actually solve the compromise?
So it's a multiplexed protein imaging technology that totally shatters that 2 or 3 color limit. CODEX-CNS allows researchers to look at 32 different proteins simultaneously.
32.
Yeah, 32 on a single, completely intact slice of brain tissue. So instead of just seeing a generic cell, they are simultaneously tagging 32 distinct features.
That's incredible.
They can identify the neurons, the support cells like astrocytes, the blood vessels, the immune responders, and the toxic disease markers. And they see all of it in their exact original architectural locations.
And there is a detail in the methodology that I found just incredibly striking. They did all of this on FFPE samples.
Yes, formalin-fixed paraffin-embedded tissues.
Right, which I know sounds like incredibly dense jargon, but for anyone who has ever spent time in a pathology lab, that acronym is huge.
Oh, it's a game changer.
Because these aren't fresh, specially preserved samples that require some, multi-million-dollar cryo facility. These are standard, wax-embedded, archived brain samples, the kind that are currently just sitting in cardboard boxes and brain banks all over the world.
Exactly. It democratizes the entire process.
Well, wait, hold on. If this CODEX technology is so powerful and the samples are literally just sitting there in the archives right now, why are we only doing this now? Like, what was stopping someone from looking at 32 proteins on these blocks 10 years ago?
It comes down to a really stubborn biological roadblock, which is autofluorescence.
Autofluorescence.
Right. Human brain tissue, especially as it ages, accumulates a specific cellular waste product called lipofuscin.
Lipofuscin, is that just like the natural wear and tear pigment?
Precisely. It's essentially undegradable junk that builds up inside the cells over decades of a person's life. Now, under normal circumstances, lipofuscin just sits there harmlessly.
Okay.
But when you put it under a high-powered microscope and hit it with the lasers required to see those glowing protein tags we talked about, lipofuscin reacts, it glows.
Oh, I see.
And it glows incredibly brightly across almost the entire spectrum of light.
So it basically blinds the camera. It's like trying to spot the faint, delicate blink of a single firefly, but you're attempting to do it while all the stadium floodlights are turned on and shining directly into your eyes.
That is the perfect way to visualize it. The intrinsic glowing noise of the aging brain tissue completely drowns out the faint signal of the delicate proteins you're actually trying to study.
Wow.
And scientists have tried everything. They've tried mathematical algorithms to subtract the glow. They've tried different dyes. But lipofuscin is notoriously difficult to work around.
So how did this specific team manage to, turn off the stadium lights?
Through a chemical fix that is shockingly elegant in its simplicity, really. They developed an autofluorescence reduction treatment. Before they even start the high-tech 32-protein imaging process, they take this incredibly thin, fragile slice of archived brain tissue, and they bathe it in a 4.5% hydrogen peroxide solution.
Wait, really?
Yeah, and while it's in this H2O2 bath, they expose it to a broad-spectrum LED light.
They literally just soak it in hydrogen peroxide and shine a bright light on it. It sounds like they're just bleaching the tissue.
Well, in a highly controlled way, that's exactly what they are doing. What's fascinating here is that specific combination of hydrogen peroxide and intense LED light quenches the lipofuscin. It permanently shuts off the chemical bonds that cause the glow.
That's wild.
But the real genius is that they found the exact concentration and duration that turns off the background noise without destroying the structural integrity of the 32 delicate proteins they actually want to study right afterward.
That is a massive operational leap.
It really is.
So just to do a quick recap for you listening, we've gone from old methods that lost the brain's spatial map entirely to this CODEX-CNS tech that keeps the map and reads 32 proteins at once. And researchers unlock this by using H2O2 and LED light to shut off the brain's natural glowing noise, that lipofuscin.
Exactly.
So here's a question to keep in mind. Imagine being able to see 32 features of a cell simultaneously. What kind of patterns do you think you'd start to notice? Because once they turned off that glowing visual noise and actually looked through this new 32 protein camera, the sheer volume of what they saw is staggering.
The scale of the data is where this stops being just a cool imaging trick and starts becoming a fundamental shift in neuroscience. I mean, to make sense of the images, they obviously couldn't just have grad students counting cells by hand.
No, that would take lifetimes.
Right. So they ran the data through a specialized machine learning platform called Ariadne.ai, and they managed to segment over 700,000 individual cells across their initial 16 brain samples.
Over 700,000 cells. And I really want to emphasize what the word segment means in this specific context, because usually when algorithms are taught to count cells, they take the easy way out.
Yeah, they really do.
They just look for the nucleus, right? The dense, round center dot of the cell. Because it's high contrast, it's easy for a computer to spot. But brain cells, particularly the immune cells, aren't just round dots.
Not at all.
They are sprawling. They have branches and tentacles and complex, irregular bodies.
Right. A brain cell interacting with its environment looks much more like a root system than a marble. And this machine learning model was trained to map the actual physical boundary of the entire cell.
Wow.
Not just the nucleus, but the cytoplasm and all the sprawling processes. It gave them a mathematically precise map of the physical shape of over 700,000 cells.
Which naturally brings us to the myeloid cells, the immune cells of the brain. When researchers started categorizing the physical shapes, or, the morphologies of these immune cells, they realized the cells naturally clustered into distinct physical groups.
Yeah, they found three primary architectures. You have a rounded shape, which is exactly what it sounds like. You have an intermediate shape, and then you have a ramified shape.
Ramified.
Yeah, ramified just means highly branched, like a complex tree canopy. And that ramified group was so complex, they actually had to break it down into 3 sublevels of branching.
And this is where the map really starts to reveal its secrets. Because when you can finally see the whole forest, you suddenly realize that a cell's physical shape correlates perfectly with the neighborhood it lives in. Form follows location, like the rounded immune cells. The algorithm showed they were almost exclusively hanging out right next to the brain's blood vessels.
Which makes perfect biological sense. A rounded cell is streamlined. It's near a blood vessel because it might need to mobilize quickly to slip into or out of the bloodstream or to interact with the circulatory barrier.
While the ramified cells, the ones with those long, complex branching tentacles, they were found deep in the brain tissue. And they were specifically positioning themselves near dendrites. And we know this because CODEX-CNS allowed the researchers to simultaneously tag a protein called MAP2, which is essentially the structural scaffolding of those neural receiving branches.
Think about what those branches are doing. A ramified immune cell is basically casting a wide net. It uses those tentacles to constantly taste and monitor the synaptic environment, making sure the connections between neurons are healthy, clearing out daily metabolic waste.
Right.
It needs those long branches to touch as many neural connections as possible without having to physically move its main body.
It's incredible. It's like looking at a satellite map and realizing that everyone wearing a life jacket is gathered near the rivers and everyone wearing climbing gear is congregated by the cliffs.
Exactly.
The physical equipment matches the immediate environment perfectly. But we have to remember, we aren't just looking at the pristine landscape of a healthy brain here. This is an Alzheimer's study. The environment isn't safe.
No, it's actively toxic. The defining pathological feature of Alzheimer's disease is the accumulation of amyloid-β, or A-beta, plaques.
Right.
These are dense extracellular clumps of misfolded proteins that build up between the neurons.
And when the researchers took this CODEX-CNS map and zoomed in on the immediate microscopic neighborhood surrounding these dense A-beta plaques, they documented something genuinely alarming happening to the local architecture.
Because they could look at 32 proteins at once, they specifically filtered for normal synaptic proteins, things like PSD95 and synaptophysin.
And we should definitely clarify what these are the literal bridges of memory.
Yes.
These proteins are the molecular machinery that allow one neuron to send a signal to the next. They are the physical basis of thought.
Exactly. In a healthy brain section, these synaptic proteins are distributed everywhere. It's this dense, sparkling web of communication. But when you look at the immediate micro-vicinity of these dense beta plaques, the web is just gone. The synapses disappear, completely wiped out. And it's not just an empty space left behind. In place of those healthy synapses, the researchers found a high concentration of what are called dystrophic neurites.
Which, I gotta say, is a very polite clinical term for a microscopic disaster zone.
That's a very good way to put it, yeah. Dystrophic neurites are essentially swollen, abnormal, dead-end tangles of neuronal wiring.
Ouch.
The toxic environment created by the plaque disrupts the neuron's internal transport system. The proteins and supplies the neuron is trying to send down its branches hit a roadblock, and they just pile up, causing the branch to swell and twist.
It shows us that these plaques aren't just, inert rocks sitting harmlessly in the brain tissue. They are actively radiating toxicity, literally destroying the delicate synaptic connections right next to them.
Yeah.
It completely reframes Alzheimer's disease. It's not a uniform fog of memory loss. It creates highly localized, specific microzones of destruction.
Right.
It's a toxic spill site. And if you are a healthy synapse sitting 10 micrometers too close to it, your structural machinery just gets dismantled.
Which brings us back to those immune cells. We know that the shape of an immune cell changes based on its neighborhood.
Mm-hmm.
But here, the neighborhood is a toxic spill. So what did the immune cells actually do?
And here's where it gets really interesting. This is the ultimate biological mystery. If an environment is shaping the cell, and the cell is trying to shape the environment, who is actually winning?
Right.
So just to recap the second part for you, machine learning mapped the exact shapes of over 700,000 cells, proving that a cell's shape correlates tightly with its neighborhood. But dense A-beta plaques act like toxic spill sites, destroying normal synaptic connections.
Yeah, wiping them out.
So the question for you to ponder is this. If a cell changes its physical shape based on who it is sitting next to, does the cell shape the neighborhood or does the neighborhood shape the cell?
It's a great question.
Because to figure this out, the researchers realized they needed to go beyond just looking at the physical silhouettes of these cells. A shape can only tell you so much. They needed to interrogate the cells directly. They needed to read their protein name tags.
Right. And because of the 32 protein panel, they could do exactly that. By analyzing the complex combination of proteins expressed on the surface of these myeloid cells, they found they could categorize them into distinct functional identities.
Okay.
They identified monocytes, different types of macrophages, but the most critical group for this story emerged when they applied a brilliant spatial analysis technique. They established a 30 micrometer rule.
Okay, walk us through the geometry of this 30 micrometer rule.
Yeah.
What does that actually look like in practice?
So 30 micrometers is roughly half the width of a human hair. It represents the immediate physical interaction zone of a cell. The researchers programmed the system to draw a tiny, invisible 30 micrometer radius around the center of every single immune cell they had mapped.
Wow, okay.
And then they asked the computer a very simple question. What else is sitting inside this specific circle?
So they were systematically checking the immediate neighbors of hundreds of thousands of cells.
Precisely. And when they ran that spatial query against the dense A-beta plaques, those toxic spill sites, they found a hidden, highly specific subpopulation of cells.
Oh, wow.
It was a subset of a group called BLM, or border-associated macrophage-like cells. But these particular cells were doing something extraordinary. In the Alzheimer's patients, they were almost exclusively swarming the plaques.
The researchers actually designated this specific cluster with a new name, right? HPM, human plaque-associated microglia.
HPM. And we know exactly what an HPM cell's functional identity is because we can read its protein name tag. They express a very specific signature.
Right.
They're positive for markers like CD163, HLA-DR, and CD11C. But there are two proteins in particular that tell the real story here, and that's CD68 and TMAM119.
Let's break those down because this is where we actually see the gears of the immune system turning. Let's start with TMAM119. What does that protein normally do?
TMAM119 is what we call a homeostatic marker. It's the protein equivalent of a security guard's badge when they are on a quiet, routine patrol. Normal, healthy microglia express high levels of TMAM119 while they're just doing their everyday maintenance in a healthy brain.
But the HPM cells swarming the toxic plaques, they have very low levels of TM119.
Exactly. They have thrown away the badge.
Wow.
The low TMAM119 tells us that these cells have fundamentally abandoned their resting state. They are no longer on routine patrol. They have shifted their biological programming entirely to respond to a crisis.
And that crisis response brings us to the second crucial protein, which is CD68.
Right. If TMAM119 is the resting badge, CD68 is the heavy machinery. CD68 is a protein that is localized in the lysosomes of a cell.
And the lysosome is essentially the cell's stomach, right?
Exactly. It's the compartment used to digest cellular debris. When a cell ramps up its expression of CD68, it means it is actively trying to eat anything large and problematic. It is the biological equivalent of a heavy-duty garbage disposal.
And the researchers found that these HPM cells, the ones physically hugging the plaques, have incredibly high levels of CD68, even higher than similar cells occasionally found in healthy brains.
Right.
So we aren't just looking at immune cells that happen to be floating near a plaque. We are looking at a highly specialized hazmat team.
Yes.
They have dropped their routine patrol gear, they have ramped up their heavy-duty digestion machinery, and they are aggressively swarming the exact site of the toxic A-beta spill, actively trying to consume it.
The precision of that insight is exactly what makes CODEX-CNS so revolutionary. We can see the crisis,
Right.
and we can see the exact biological state of the first responders.
But as clarifying as this map is, it puts us in a really fascinating position regarding time.
How so?
Well, the spatial map is a snapshot. It gives us incredible detail of one exact moment, but it doesn't give us the timeline, because the study notes that a few of these HPM-like cells, these specialized hazmat workers, they do exist in healthy, aging brains too, even before massive plaques form.
I see where you're going.
Right.
So which comes first? Are these just normal aging immune cells that sense a microscopic toxic plaque beginning to form and they migrate over to try and clean it up? Or do these specific cells get sick, break down, and then the toxic plaques actually crystallize around their failing bodies? This raises an important question, and it is the ultimate correlation versus causation dilemma playing out on a microscopic scale. Are they the cause of the fire, or are they the firefighters who arrived at the scene but got overwhelmed?
Right.
We now know exactly where they are and exactly what gear they are wearing, but the sequence of events that led to that standoff is still a mystery. It is undeniably the next great frontier in Alzheimer's research.
So just to do one final mini recap by looking at a 30 micrometer radius, researchers found a unique cell cluster called HPM. And these HPM cells have a unique protein signature, high CD68 and low TMAM119, and they swarm dense A-beta plaques in Alzheimer's brains. So, for you listening, think about this. If you were designing a drug, would you want it to attack the plaque directly? Or would you try to boost the HPM hazmat team that's trying to clean it up? because that mystery fundamentally changes how we have to think about treating this disease.
It really does.
For decades, the pharmaceutical strategy for Alzheimer's has often relied on what we might call a sledgehammer. Well, actually, a better way to look at it is like dropping fire retardant over an entire forest.
Yeah, systemic treatments.
Right. We design a drug, flush it through the entire brain, and hope it puts out the fires we can't clearly see, while hoping it doesn't damage the healthy trees.
But when you understand the exact state of a single cell in a highly specific 30-micrometer location, that overarching strategy seems incredibly blunt.
Exactly. If you were a pharmaceutical scientist looking at this CODEX map today, you wouldn't drop fire retardant on the whole forest. You would design a drug that acts like a highly specialized radio signal, a signal tuned exclusively to the HPM hazmat teams.
Right, a sniper approach.
A drug that specifically binds to that unique CD163 or CD11C protein signature, telling those specific cells to ramp up their CD68 garbage disposals even further, giving them the energy to actually clear the plaque, while leaving every normal resting cell in the healthy parts of the brain completely untouched.
It moves us toward true precision medicine in neurology. And if we connect this to the bigger picture, that really leads into the most profound implication of this entire study.
Yeah.
We started by trying to map a disease, but what we've actually uncovered is a fundamental rule of cellular biology. A cell's identity, its physical branching shape, its protein name tags, its very function is intrinsically tied to its microneighborhood. The environment dictates the cell just as much as the cell dictates the environment.
It makes you wonder, if our cells fundamentally change their identity based entirely on who they are sitting next to, what does that mean for how we approach other localized brain crises?
Exactly.
Like if someone suffers a microscopic stroke or has a small localized brain tumor.
Could the future of neurological medicine be less about trying to forcefully inject therapies into a sick cell and more about finding ways to renovate the neighborhood around it?
Oh, wow.
Yeah, if we can pharmaceutically clean up the microenvironment, clear the local toxic spill, does the HPM cell naturally put its resting badge back on? Does it heal itself just because the neighborhood improved?
It is a profound paradigm shift. From obsessing over how to fix a broken tree to realizing we might just need to heal the soil around its roots. Well, that is all the time we have for today's deep dive. We started with a decades-old scientific frustration choosing between the map and the details, and we ended up watching a microscopic hazmat team fighting a localized toxic spill in brilliant 32 protein color. Keep looking closely at the world around you, never stop questioning the accepted boundaries of science, and we'll catch you on the next deep dive.